asked me to. Got it. It's recording now. So thank you, David, and happy Veterans Day to everyone that's taken the time to join today. I'm really happy to be able to share the Regional Shellfish Seed Biosecurity Program with the U.S. Aquaculture Association through this webinar series with all of you. We've been working on this problem for a while and have moved it forward as a collaboration of industry members, scientists, regulars, and extension personnel. It's really been a grassroots effort rather than a top-down process to develop methods that protect the shellfish aquaculture industry and natural resources from shellfish pathogens. Our focus is to use the best available science to minimize risks associated with the movement of shellfish, in this case, shellfish seed within and among bodies of water. But it's really been the transfer among states and the diversity of regulations across states that inadvertently inhibit the safe development and expansion of the industry while trying to minimize risk from disease that we're trying to resolve. Okay, couldn't get my slide to advance, sir. I'd be remiss if I didn't acknowledge all the folks that have worked hard to help develop this program. It is, as I said, a grassroots effort. We have quite a large team here. You can see at the top of the slide here, and I'm not going to read everybody's name off. You can do that. And quite a number of partners and agencies that are supporting it, either financially or in kind in one way or another. I should note that the program is still developing and it's going to continue to evolve as new information and resources become available. So what I'm presenting here is the current state of the program. And if you have input, we're happy to receive it. You can contact any of us at any time. So to get into it, provide a little bit of background. As most of you know, molluscan aquaculture has been expanding along our coasts. We may not be leading the world in production, but there really is significant potential for growth in what is already an important industry in many coastal communities. We know all too well that there are many impediments to the growth and that there are a variety of efforts to understand where and how aquaculture can grow without compromising other coastal activities or threatening natural resources that may still support wild fisheries or are being restored for their inherent ecological value. One of the major risks or threats to aquaculture or even from aquaculture is the potential to spread or exacerbate disease. There are quite a few shellfish pathogens that are well known to cause mortality in both wild and farm populations, such as those shown here for the East Coast. There's a logical emphasis in wanting to understand how to stop these from spreading and exacerbate problems, but the concern given to some of the more ubiquitous ones like Dermo may overstate actual risks. And as a result, because time and resources are always limited, overemphasis on diseases such as Dermo may actually limit resources available to address emerging or unknown external risks, such as OSHV, which has been disastrous in other parts of the world, but not yet here, and hopefully not ever here. So regardless of all this, the question is, how do we minimize the risk of transferring shellfish or transferring disease or introducing disease with shellfish seed moved for aquaculture or moved for restoration or even enhancement of fisheries? We don't want these things spreading around any further than they are, and we don't want to see any new ones moving in. So more than 2 decades ago, there was a workshop in Charleston, South Carolina, that addressed these very issues. Because South Carolina and other states were becoming concerned about the widespread movement of shellfish, mostly clams, particularly hard clams at that time, from northern to southern states and back again. They were trying to take advantage of longer growing seasons, and the movement was at one point linked to QPX outbreaks in hard clams, which had devastating effects in the northern areas. So this workshop happened, and they completed a review that revealed a variety of approaches to biosecurity with varying levels of risk. But the more generally accepted approaches are summarized here on this slide. The workshop recommended better, more informed coordination, the development of a surveillance system, excuse me, and a web-based system to share the data. They recommended improvements in standardization in the assays and detection methods, and a variety of other mechanisms to improve biosecurity, with an eye towards not compromising commerce. So you don't want to be so fearful that you shut everything down and say nobody can move anything anywhere, but you also don't want to just let people move things willy-nilly. You have to have some sort of regulation on it and some sort of control. So despite these recommendations back in 2002, not really much has happened, most likely due to resource limitations and workloads. And at the time, shellfish aquaculture was not really all that large of an industry. There was a lot of clam aquaculture going on, but not too much in oysters or other shellfish, scallops, and whatnot. That's all changing. It's changing quite quickly. This status quo that's remained has also kept problems that were there, and those are kind of outlined here. There's a piecemeal surveillance that happens, and this continued with limited scope. It's often tied to research projects or specific fisheries interests, and there's no real coordinated database to access any of the data that comes out of that. This had both a direct and an indirect cost in terms of time and money, as the industry has continued to expand the capacity to process seed, transfer health exams, and is outpacing the capacity to process those samples. At the federal level, the USDA, for example, has only 2 labs that they've certified to perform shellfish health exams, one on the West Coast, one on the East Coast, and no current mechanism to certify other labs. So most of this is done by university labs or 1 or 2 commercial labs that have expertise in pathology, and they do that for the states. And there's no coordination among us. So this leads to things like delays that are problematic, particularly when risk is low and transfer could move quickly, or in an area where seed continues to grow while the assay is being performed, that potentially exposes it to additional pathogens as it waits for approval to be moved. So what's being examined is a different product than necessarily what's been being moved, based solely on how much exposure it's had to disease. Another problem is that state jurisdictions don't always align with pathogen distributions. You can see that in the maps shown here. So while moving shellfish from one side of, say, Delaware Bay to the other side is often of little or no concern because the pathogens are all mixing throughout the bay, it may require costly evaluations for approval that are not in line with the risks. This leads to zero tolerance policies that develop naturally in the absence of information. And they can unnecessarily stifle commerce as well as fisheries enhancement and restoration. So it behooves us to find a better path forward. With that in mind, we've created the Regional Shellfish Seed Biosecurity Program, or the RSSBP. Now, I know that's not necessarily an acronym that's going to roll off your tongue, but the name clearly conveys what it's for. The program's actually meant to be national, but it's being built regionally and will always operate regionally because the shellfish and the pathogens that are present in different regions are quite simply different. There are different concerns in different regions, and each region operates independently from the other as a result. We're not in this program, for example, advocating to move seed or recommending the move of seed, particularly along coasts. Our goal is simply to enhance biosecurity in those instances when seed will be moved, and we anticipate movements will continue to increase with the growth of the shellfish aquaculture industry. And also, as well, with the growth of interest in shellfish restoration, as well as the growth and interest of restoring fisheries, so enhancing what fisheries remain. An added benefit of the program is efficiency, which helps reduce time and expense. So we've employed a process that's collaborative and inclusive, beginning with the East Coast in 2014. We're now working in the Gulf Coast and have plans on expanding the program to the West Coast. Our focus has been on developing recommendations, many of which were identified in the 2002 document, as well as tools needed to provide clear and effective strategies that are grounded in science, so that precautionary measures are based on logic and evidence rather than fear of the unknown. The RSSBP has 4 core elements, which I'll discuss for the remainder of the talk. These first 2 I can knock out relatively quickly here, and then I'll focus more on the other 2. The first one is Actually, the first two are committees. The first one provides stakeholder input, the Regional Shellfish Health Advisory Council. And the second one, the Regional Network of Shellfish Pathologists, provides us with the expertise needed to understand what's happening with any pathogens. This is our current advisory council. It was first created in Portland, Maine during a workshop of more than 50 participants from all along the East Coast. Membership is designed to represent the sectors of industry, regulation, pathology, and extension. You can see that under the areas listed here. Presently, we have both the East and Gulf Coast represented, although it's biased towards the East Coast because that's where we started. As the RSSBP expands, each coastal region may need to create its own advisory council, or we may need to expand the size of this council or simply just redistribute the membership to be more geographically represented. Whatever we do, we're going to adjust it and adapt it as needed to meet the needs of the program. To date, this council has functioned quite well as both a resource for the RSSBP and also for helping resolve uncertainty for regulators about proposed transfers. So in these cases, the Advisory Council works with the Pathology Working Group. They lean on the Pathology Working Group on their expertise where needed. I don't have a slide for the Pathology Working Group, but there's not a lot of shellfish pathologists out there, so we've taken the liberty of basically adding all the active shellfish pathologists we know to that working group, and we'll continue to do so as we expand the program, taking in pathologists from the Gulf and ultimately from the West Coast as well. And thankfully, pretty much everybody's been very open and receptive to helping us lead this program and provide their expertise whenever they can. So what I'd like to do next is to show you our interactive disease database, but I don't know if this is a good time to pause for a second, if there are any questions. Does that sound like a good idea? Scott, not Scott, David?
Nope. Well, I don't see any questions typed into the chat box. So let's continue, and then we'll have folks hold questions to the end. Okay,
I'll probably stop once more between these sections because there will be a lot of information I'll be presenting next. So I will have to admit that I'm not quite technically savvy enough to jump from a PowerPoint to a live database. So I'm going to show you still images of some of the things that the database can display, and then I'll invite you to explore it on your own later. So let's get right onto the database. All right, we call this the Shellfish Disease Mapper. It's an interactive map that's linked to the database. It allows you to examine and compare pathogen distributions, pathogen profiles, and pathogen histories in relation to seed sources, or just in relation to where you're located, whether it's a farm you're operating or whether it's an embayment that you're working in. You can zoom in wherever you want. The homepage calls up this map that gets you started. It's focused on the East Coast because that's where we began. But you can use the Find Location button. I don't know if my mouse is showing up or not, but there's a Find Location tool over here. And you can type in a Latin lawn or a place name. That can be a city or a town, or it can be a body of water or a river, and it will zoom you into that location. You can also, if you like to, there's a thing here for the base map. If you don't like this particular map, you can put in one with streets in it. You can put in one that's a satellite image. There's a variety of options there, and you just pick whatever your preference is for the base layer to help you find things. At any point in time, there's this Save Share Print tool, and so you can use that. So let's say you're working to establish a shellfish lease, and you're working with the state regulator, and you want to show them where you're doing it and what these things are. You can make a temporary map that will give you a quick URL that you can share with them, and they can look. It'll pull it up right on their screen if you share that URL with them. You can save the map, which will give you a permanent one. So you could do that. Maybe you're working in the evening and tomorrow you want to share it with them. You can do that. Or you can print it out and you walk into their office and carry the map in with you, or mark it up, whatever you need to do. So that's kind of a nice option that's on here. If you are a bit more advanced in your understanding of GIS tools, there's also an import data function here. So perhaps you want to import the state's data for your local shellfish classification waters, or where marinas are, or boat launches, or whatever data you might have. You can import that data into this session that you're working on. I would note that whenever you import data, that data is specific to the session that you are running, and only available to you while you're running it. Once you turn it off, that data will go away and exit the mapper, so that you're not putting in data that you may not want to distribute to anybody who opens it up. The real meat of the tool, however, is in the data, the disease data, which are most easily accessed by adding map layers. That tool right over here. Again, I'm hoping my mouse is showing up on the screen. And so let's take a look at some of the data displays. Okay, here, using map layers, I've turned on Dermo disease for oysters and asked it to show us data from July to December. That's the period, at least in this region, when Dermo levels are typically highest during the year. I've turned on the legend. So there's a legend over here you see kind of grayed out, and this is the legend here. So the legend shows a scale of prevalence, which is a percent of animals that are infected. And so if it's down around that greenish color, there's no animals infected. If it's in the red, you've got 100% of the animals infected. When you're selecting data to display, you can pick particular years or particular periods of times. Default selections are the first year, or the first or second half of the year, or the entire year. You can select the current year, or you can select the most recent 3 or 5 year period, or you can custom add a period of time. The map displayed here shows all the data that have been entered into the database, so it's data from all time. I've clicked on a point in Delaware Bay for this display, and it puts that pop-up in here that shows you we're in an area of Delaware Bay called the high mortality region. And it's showing data from 2014 to 2021. And it's giving you a little bit more than just the prevalence. It's also giving you the total number of samples. And in that period of time, there were over 5,000 samples. It's giving you something called an advanced prevalence, which is a percent of individuals with heavy infections. And it's giving you something called a weighted prevalence, which is an average intensity of those infections. And so depending upon your knowledge of diseases, that data may be meaningful to you, or you may be just satisfied knowing the percent of animals that are infected, which is the prevalence. Sometimes it's easier to understand the data in more of a graphical output. So there are various graphical outputs that are available. Here in the upper plots, we're showing annual patterns. And so they're indicating the annual pattern of prevalence, the advanced prevalence, and the weighted prevalence. They usually track each other, and you can kind of see that in these plots. Those 3 metrics track each other. The lower plots are showing you seasonal patterns. So you have months of the year here, and so they're averaged for the entire month of the year. And this is showing you just, it says low prevalence, but that's just the absolute prevalence versus the advanced prevalence, the heavier infections. So the total prevalence is higher than the advanced prevalence, which should always be the case, unless everything is advanced. And you see that this is the typical pattern we see in Delaware Bay, an increase across the summer to a peak in the fall, and then it starts to decline. Same thing for the intensity, the weighted prevalence here. And then over here, it's the same data, but it's put into a box and whisker plot. So those of you who are a little bit more statistically minded may want to look at the data in this kind of fashion. That gives you an idea what the variability is in the data set. You can download all these data as well. I should mention that also, if you need to. You can have it print out reports that summarizes the data for you. This is a summary for Delaware Bay, New Jersey. I'm not sure why I did this, but it also has the state here, which matches that. So I think there was a little glitch in it when I did this. But when it does state statistics, it's telling you all the diseases that are known to be possibly present in the state, and the species that are known to exist in the state in different environments. And that gives you the prevalence statistics down here. What's actually the most useful, at least for the way that we've been operating this tool, are site comparisons. So maybe you want to know where you're going to purchase your seed from and where it's going to be deployed. What's the source condition versus the destination condition? This allows us to do things that are what we call like-to-like decisions. And so what I mean by like-to-like decision is here, if the disease at the source is less than or equal to the disease at the destination, we call that a like-to-like transfer. So you're not really going to do anything to exacerbate the problem. You're not introducing anything new. On the other hand, if the source has a greater incidence of disease than the destination does, then you're basically moving more heavily infected animals to a less heavily infected area, and you may not want to do that. And so these kinds of comparisons allow you to make a decision about what might be the benefit or not of such a transfer. Here I've shown just Virginia as a whole versus New Jersey as a whole, just for simplicity. But in actuality, you probably want to pick specific sites that are more zoned into somewhere that you want to move things to. You can also compare these graphically, as I showed in the previous slide. So I'll show you that here, because sometimes comparing things with numbers can be difficult, whereas visual display of it is sometimes more easily accepted by people. So here you can see that the lines here represent the different states, and you can see that they're really kind of tracking each other fairly well. Although here Virginia did not have many advanced prevalences, New Jersey does. So if you're going to move from Virginia to New Jersey, that would not be a problem. But the other way maybe would not be so preferable to do. And you can also look at the other plots that we discussed earlier here as well to see how things are changing seasonally within each area. This kind of information allows you to identify, oh, what's the risk if I'm going to do this transfer, say, in the springtime versus if I'm going to do it in the fall. So obviously in the fall, animals are more heavily infected. You're going to be moving more heavily infected animals than if you move animals in the spring. So these data allow you to look at the data and make those sorts of inferences that might help you make a decision about the biosecurity of any particular move or transfer. Okay, so as I said, David, and to the audience here, I was going to stop at another point and maybe see if there are any questions on the database. I see there's something in the chat, looks like.
Yeah, I just put the link to the database.
Okay.
I was looking, when I pulled it up, David, I don't see a way to start the program that you're in. Is that something you have to download, or is that?
The link might not be active right now. I'd have to check with our programmer Lucas Markson to see if it's active. But on the disease mapper page, if you scroll down, there should be a link there. If it says it's not up yet,
Yeah, it says it's under construction.
Oh, okay. It's constructed. You can see the things I was showing you there. He just has it down probably for some maintenance, and hopefully it'll be up very shortly. I won't promise it during this talk, but
Very good. I'm glad you did your slide captures, screen captures. I don't see any other messages, so I think you're free to proceed.
Okay. All right, so if we think back to that 2002 workshop, and I don't know if John Croyder is on here, but a shout out to John Croyder for an excellent job pulling that report together so many years ago. We still use it as a guideline. But that survey had indicated that 8 states actually reported back then that they had a process to approve hatcheries for shipping seed into their state. The methods that each state used were not consistent, and they largely relied on the experience of the state regulators with those hatcheries and the relationships with those hatcheries. So they varied from state to state, and there wasn't any. Each state might have had some guidelines of things they looked for, but they weren't standardized in any kind of way. So what we've done here is try to build upon that and develop a set of best management practices for hatcheries, for hatchery biosecurity, and created a program to evaluate whether or not the hatcheries are being compliant so that we can help build confidence for the regulators in a way that also promotes hatchery success, because a hatchery wants to produce healthy seed. It's in their best interest to do that for their customers as well. You know, if they're sending you sick seed, you're not going to continue to buy from them. So what can they do to improve their biosecurity? So I know a couple of my colleagues that have been working on this are here. Karen Hudson in particular worked really hard with Lisa Calvo to put this this Biosecurity Hatchery Compliance Program together, and it's available on our website. We do have, should have mentioned this at the beginning of the talk, we all have this code here. I think it'll work if you click on it, but if not, our address is pretty simple, rssbp.org. And you can download this manual. It's not very long. It's pretty logical and straightforward. The idea is to encourage the movement and the sale of the most biosecure products whenever possible by using best management practices that minimize the occurrence of disease pathogens in the hatchery. The program also creates a documented health history for the hatchery that helps them understand and improve their operations so they can produce the healthiest products possible. And that provides confidence to the buyers, but more importantly, to the regulators that are going to approve a transfer in those products. So in this way, it enhances biosecurity, which should facilitate decision-making and ultimately save time and reduce costs for everyone. So the premise of this is that smaller products are more biosecure. And, you know, this is what really underpins this program. Quite simply, younger animals have had less time to contract disease and are more likely to have been protected from exposure while they're small and maintained on treated water within the hatchery and fed cultured microalgae. So they should be, just based on that, theoretically less likely to be carrying disease. This means the most biosecure hatchery products are likely going to be larvae and newly set seed. Once they move out of a hatchery, the risk of exposure increases as with the amount of time that they're on raw water and exposed to whatever pathogens are circulating in the surrounding waters of that hatchery. So in this graphic here, we're sort of showing that, you know, green is good, red is bad in terms of disease risk. And so the smaller you are, the better you are. And then along the bottom here, so here's some life stages shown, some larvae, some small, maybe 2 to 4 millimeter seed, some larger seed that have been out in a nursery, and then some broodstock and adult oysters that are ready for market. And a little bit of data from my colleague Ryan Carnegie at VIMS here from their lab. This is from a 2-year period of 2017 to 2019. Showing their results for Dermo and PCR of this, where they really have never detected any Dermo cells in larvae. And then looking at seed, in this case, I have both Dermo and MSX shown here. So these are 41 samples. A sample consists of 60 or more individuals, and never detecting either MSX or Dermo in these small things that have not left the hatchery yet. But then once they move out in the field, we didn't see much MSX yet at this maybe 10 to 15 millimeter stage, but they're seeing, you know, an increase in the amount of derma that's detected. Now, remember, a sample I said is 60 or more animal individuals, and so a detection of one of these means maybe one animal of those 60 had a derma cell in it. So it's still a really low infection level, but nonetheless, they're starting to build up infections as they move. So again, it's shown that this is yellow up here because while there's an increased risk, it's still not that high until you get out to the larger mature adult animals. And just look at my notes here to see if I missed anything in that talk. No, okay. All right, so so with those principles and data forming the basis of our best management practices, that's what we've set up in here and designed to keep pathogens away from to keep hatcheries biosecure. All right, so the best management practices are designed to keep pathogens away from larval cultures and keep them away from seed while they're in the hatchery. The BMPs are detailed in our manual, but there's only 12 of them actually, and they're summarized on our website, and they boil down to 12 actions They're listed here, but I'll show you a briefer description of them here and walk through them a little bit. So they're all pretty much common sense. When you're in the hatchery, you want to separate any potentially contaminated animals, which would be your broodstock, from your biosecure animals. That's basically your larvae and your seed. You want to separate areas that are using untreated water, the raw water that might be conditioning your broodstock to spawn, from areas where you're using treated water, where your larval cultures are. Treated water requires that it be filtered down to 1 micron or some other means that minimizes disease exposure. So other means might be something like pasteurizing the water, or using well water, something like that, or even some hatcheries actually make up their water. Cleaning of potentially contaminated materials, that should be done away from any cultivation that's happening, so that you're not washing things off and splashing them back into a larval culture. Records need to be kept of maintenance, particularly of equipment that needs to be, filters need to be changed, and those sorts of things. You need to document and follow the workflow and have an operational plan that helps prevent contamination, so that you're not having yourself or other workers move from contaminated areas into uncontaminated areas on a routine basis, or some sort of process there to prevent them carrying anything back and forth. And that would include things like avoiding sharing equipment among those areas, or develop a sanitation protocol between them. So if something's going to get moved from one area to another, maybe it needs to be washed first. You should be performing health examinations whenever you have a problem of mortality. So whenever larvae or seed, if they stop growing and they start dying, you should start thinking about having health examinations performed, so you can identify what the problem is as quickly as possible. One of the big failures as a shellfish pathologist that happens is we'll get samples sent to us after the mortality has happened. And so what remains are healthy individuals, and doesn't really help us identify what the cause was as we look at the ones that weren't impacted. Maintaining records of the broodstock that document the source, the genetic background, and the collection date. So where are those from? Maybe they're the source of a problem that you had. Maintaining spawning records that document the broodstock, again, that was used, the date they spawned, and other info, so that if you want to have any kind of traceback study to figure out what the problem was so you can fix it, that information is available. And then if applicable, if you're using, let's say somebody contracts your hatchery to spawn their local broodstock, but you're in another state, they'll ship you their broodstock. You should have a quarantine system to handle those broodstock that prevents anything that they're carrying with them from getting into your local waters, if you do that. And then lastly, you need to follow all state permitting requirements. So those are basically our best management practices. I see a couple things in the chat. Let me just look real quick. Oh, those were.
I'm just sharing pages as you're going through them, but we did have one question ask about if there are any plans to include Pacific oyster surveillance in the database.
There are, but we have not moved to the West Coast yet. So as soon as we do that, the surveillance data would be there. And the data I shared was just for oysters, but we do surveillance of hard clams, scallops, whatever is being cultured. Most of the focus on wild population surveillance has been on oysters, and so the database is dominated by that right now. But any surveillance data for any shellfish species would be can be entered in the database. All right, so moving along on here for the Hatchery Compliance Program, we are always emphasizing that this is a voluntary program. So presently there is a system that people can follow where every time you ship a batch of seed out, you get it certified, you pay for that certification, you wait until it comes back, and then you can ship it out. If you're in this program, one of the ideas is that you will already have done some of those pathology exams, and you have a track record of producing clean stuff, and a regulator can see that track record and then say, oh, this stuff is clean, you can ship it out today and not have to wait for an exam to happen. So makes you a bit more nimble, It makes the buyer happier, makes the seller happier. It's a lot more efficient process. But if you're going to participate, this slide then demonstrates some of the things that you're going to need to do. So if you're a hatchery, you have to demonstrate implementation of the BMPs in your hatchery. So the first thing is to develop how you're going to do that. Next is to undergo an audit of the operation. And then if you want to certify any of the products coming out, you need to have disease surveillance that maintains a rolling 3-year health history. As I get into this a little bit more, you can do the first one and wait for the second one to happen, or maybe you're not even interested in the second one happening. You just are satisfied having a biosecure hatchery. You can stop where you wish. But we would encourage everybody to do everything. States that choose to participate, they should be developing rules, regulations, or policies that permit a streamlined process for import permits when a hatchery is participating and has passed all of these things. So if the states, this is key, if the states are not accepting this, then it goes back to the status quo and everything gets bogged down again. So having the trust and the participating in the states is very important. And then we also expect the states to assist us in obtaining data that supports the database, any surveillance they're collecting, whether it's wild data or whether it's hatchery compliance data. And so we can work collaboratively on that. We're not necessarily asking them to set up a surveillance program just to share with us what they're already doing, because we know what their budgets are like. And again, if you decide not to do this, you can follow the status quo, which means batch certification for all transfers, according to whatever tolerance levels apply to the state in which you're transferring the seed to. So mind you, some states have zero tolerance regulations in place. And so if anything is detected, they'll say, sorry, you can't bring that in. And you'll have to go somewhere else to find seed that's clean. All right, so the process to do this is shown here. It's pretty straightforward. This is all available online. We have a hatchery will submit an application that will get reviewed to determine whether or not BMPs are addressed. Then we'll have an audit first of the hatchery to see if those BMPs are being implemented. And then secondly, if they want any products assessed and certified as clean, we'll review the health assessments. And then they get a decision letter that the hatchery can then use in their applications for transfers. So to get things going, the application, which is available on our site, would describe how each BMP is addressed. You'd list what species and broodstock are being held in the facility. If you identify any products that you want certified, and if that's the case, you give permission to release health evaluations. So those health evaluations come from the pathology labs, not directly from the hatchery. And so we would request to have access to them from the pathology lab. Once that's done and we've verified that the BMPs look to be adequate, we'll have an audit team comprised of independent experts. Here we have John Ewart, who some of you may know, who has developed expertise over many years working at Delaware Sea Grant. And he went through and is looking at a hatchery. I can't remember which hatchery this is, but was observing what their practices were and doing it. And you can see we were doing this during COVID. But he and others that we have agreed to help us out with this and get the program working. Their bios are listed on our websites. And so that team then sends back a report that's reviewed by the advisory council, and they can discuss any concerns and get back to the hatchery if need be, or get on to writing them an approval letter. The second aspect of the review, if you're going to have any products such as larvae or seed done, you would send samples to pathology labs. States have generally agreed that they'd like to see 3 years of samples with 2 samples per year, although some states may be more lenient than that. I don't believe any are more stringent than that demonstrate the level of pathogen detection, which was in there. It should be 0 if all things are operating properly, But as I said, there's like an overemphasis on Dermo, for example. Dermo is pretty much ubiquitous from Massachusetts south. We see it everywhere, and transferring a few cells of Perkinsus really probably is not going to make that much of a difference. So there's a little bit more leniency on pathogens such as that. But for the most part, they should be zero. This may change as the program matures and confidence builds that biosecurity for one species applies to all species. So instead of having it be species and size specific, it may be that you can test your clams at one testing, test your oysters at another point in time. And as long as they're always coming up clean along this time period, that may be satisfactory. Presently, however, it needs to be done for each species and each size that you want certified. It should be done on the largest size product that you are interested in transferring, because that will cover all earlier life stages. So in this sort of example here, this is an actual pathology report from some of our seed produced here that were, I can't remember where they were getting moved to, but in any case, we sent them some seed. And the seed came up clean. The examiner said that nothing was detected. The RTM results did not detect anything. So any larvae produced in that hatchery, because of they had to be larvae before they were seed, would also be cleared for movement. But anything larger that hasn't been looked at would not, because this has been exposed longer. And so you want to use the largest seed that you have intent of shipping out under this program being examined in these pathology reports. Following the audits, whether it's a facility audit or a facility plus a seed, a letter of compliance is sent to the hatchery. They can include that in applications for transfers, or they can send it to customers to include in their applications, whichever is appropriate. The RSSBP will post a listing of approved facilities, which is shown here, and the products from each facility that have been approved as well. So in this, we're starting to separate these out by states so people can go quickly to the state they're interested in. And so this shows you here the current status of things that we have already examined. So this first one is for Mook Sea Farms up in Maine. They've been in the program for 2 years now. It provides their address. The last facility audit was in February of this year. So the compliance period is for 2022 this calendar year. So they are currently in compliance as a biosecure facility. And here are the products that they asked us to look at. And so we looked at 1.5 millimeter and smaller seed. Actually, we looked at 1.5 millimeter seed, which conveys compliance to anything smaller than that. They have a high level of biosecurity because nothing was detected. That was audit was conducted in January. And again, here's the time period for which this is good. And if you go onto our website, you can see the other hatcheries that have gone through this process as well. And this information is readily available to the regulators also. So the compliance letter would go in the application. And then the state regulator, if they wish to, they can come onto this site and make sure that everything is up to date and tracking well. To help inform their decisions and verify anything that's in the certification letters. They don't have to track down the pathology labs to do that. They can look right here. Nurseries and growers can use these lists to help them identify biosecure sources of seed if they wish. Local seed, however, may be their first choice. And if that's the case, they may not need to do any of this, because if they're buying from somebody who's right next door to them, everything is the same and they're not transferring anything. So the two things that this program may sort of incentivize, one is buying locally, and the other is if you're going to move seed, buy things that are the most biosecure product possible. All right, and so we're now in the process of witnessing regulatory adoption of this. So Virginia, for instance, changed their actual statute in order to incorporate the use of biosecurity certifications. They didn't explicitly mention the RSSBP program because it's a statute, so it's sort of nondescript to what the program is, but it allows you to have a hatchery certified through this. And virtually every other state from Maine to Texas is now looking at how they can utilize this program. We spent a portion of the last year meeting with the regulators in each state to inform them about this program and how they can use it. We got a lot of valuable feedback from them and used it to alter things so that we were going to be sure that we were building their confidence as well. They should be using this to help make their decisions on interstate transfers now. Again, state adoption is voluntary, but they should implement it within or modify their existing regulations. Hopefully it will help streamline their management and review process. And if something doesn't pass through this, they can still continue with, and they will need to continue with the current process where appropriate. So for instance, we're not yet working with nurseries. That's the next phase that we're going to be working on. Ryan Carnegie and I and the others on the program recently submitted a new grant proposal to expand this to a nursery system. So we hope to start making progress on that as well. We don't envision that nurseries will have the same level of biosecurity as a hatchery, because as I showed earlier, the basic logic of this, as animals grow, the increase of disease risk increases. One final comment is that nationally, the USDA, through their APHIS program, that's the Animal and Plant Health Inspection Service, is developing a set of comprehensive aquatic health program standards called CAHPS for short. Their main interest is to ensure the marketability of aquatic products for international trade by ensuring that they meet international standards for biosecurity. So for instance, we have up in Maine and on the West Coast, there are hatcheries that work back and forth with programs in Canada in shipping things back and forth. And Canada is very concerned that our standards are as strict as their standards. And so APHIS has a vested interest in helping to make sure that the international trade can happen. APHIS, however, has relatively little experience in Molluscan shellfish. The animal side of them is we used to work with livestock, cows and pigs, and things that they can count and examine individually. You look at the looking at a pile of shellfish seed is a little bit foreign to many of them, and so they're looking to partner with us and help them align the RSSBP program with the CAHPS program and maybe merge them together at some point in time so that they can take advantage of the expertise that we've been building in this program and apply it to their program. And so with that, I'll thank all of you for your interest and your attention, along with David Klein and the U.S. Aquaculture Society for this opportunity. And I'm happy to address any questions that anybody might have.
All right. Well, to be respectful of everyone's time, we appreciate you attending today. And David, certainly appreciate your time and expertise and all of the people that worked on this project. With that, I just wanted to remind folks that this recording will be available on the United States Aquaculture Society YouTube channel, as well as the Aquaculture Education and More YouTube channel. So with that, I will go ahead and wrap it up.
Thank you.
Very good. Lots of thank yous coming in, David. Appreciate your time.